I'm going to talk about now for about 40 minutes or so about the inherited eye disease work that we are undertaking. As certainly those of you who have worked with me for as long as many of us can remember, will know that the Kennel Club Genetics Centre has got a very sort of long and successful track record investigating inherited eye diseases, and that I think is in very large part to this man, Keith Barnett. I suspect there's people here who knew and remember Keith Barnett. So Keith Barnett was a very eminent veterinary ophthalmologist who set up the whole small animals clinic at the Animal Health Trust, but certainly the ophthalmology unit that in its day was the largest in Europe. And when I joined the AHT in 2001, I had done a little bit of work on inherited eye diseases when I'd been in Elaine Ostrander's group at the Fred Hutchinson Cancer Research Centre in Seattle. But it was really Keith who sparked my interest in inherited eye diseases in the dog. And right up to the very end, Keith was interested in helping us study the genetics. So I don't expect to read all of this, but this is just a chart showing every line is a genetic variant that we have published on that is associated or causes an inherited disease in the dog, and everything in green is an eye disease or a condition that has an ocular component. So just goes to show that it really has been the sort of jewel in the crown of the genetics centre over the years. So we part of our success is due to the sort of close collaboration we have with the eye scheme that many of you will know all about, that's operated by the British Veterinary Association and the Kennel Club and the International Sheepdog Society. And so I'm a member of the iPanel Working Party. So there's about 35 panelists, I think, currently, although they're busy recruiting more to give better coverage over the whole of the UK. But they have a working party of about half a dozen of the ophthalmologists who are very engaged in research and very interested in doing more beyond just the routine eye exams, and I attend those meetings. So we have a very good sort of dialogue between the genetics work that we're doing and that eye scheme. So there's currently 12 different eye diseases that are listed in 65 different breeds, and they're on that chart there. So many of you will be familiar with this certificate. I think this is the latest certificate that the scheme operates. They change them quite often, and it's very busy now. And I'm quite proud to say that some of the changes that have been instigated have been a result of work from the Genetics Centre. So this box at the bottom is a tick box for all the conditions that are listed on the list of, used to be called Schedule A. There was also a Schedule B. They've both been done away with now, and now there's just this list of what they call known inherited ocular disorders, or KIODs. So if, for example, if we go back, so just to look at the top, Alaskan Malamutes for cataract. So if your dog is an Alaskan Malamute, the ophthalmologist will specifically tick the box, the hereditary cataract box, to say whether it's affected or unaffected. But then the ophthalmologists are now also, anyway, that box there is a section where the ophthalmologist is supposed to now write any other abnormalities that they note. So not just the certified diseases for the breed that they're looking at, but if there's any other abnormalities. And some of them are better at remembering to do that than others. But, for example, if the dog has entropion or something else that's not a certifiable disease, but it's something of note, they have an opportunity to write it there. Down at the bottom left, there's now a grading scheme for gonadisgenesis. So many breeds are predisposed to closed angle glaucoma, and gonadisgenesis is a risk factor for closed angle glaucoma. Oh, thank you very much.
The other way around.
So oh, thank you very much. I stand here. So yeah, this box down here is now a grading scheme. So until a few years ago, the dog would be noted as just affected or unaffected, but the gonadisgenesis can affect different degrees of the iridocorneal angle, and so we decided it would be better to collect a bit more sort of nuanced data rather than just affected and unaffected, and that really came about as a result of the work that James Oliver did for his PhD while I was certainly the AHT. He has a particular interest in inherited forms of glaucoma. Because as a veterinary ophthalmologist, they're very frustrating to treat. They're quite difficult to treat. And then the final thing is now there's a box to say whether a DNA sample was taken, and those DNA samples will come to the genetics centre. So again, the ophthalmologist, we try to encourage them to, if they have a dog in front of them that has a condition that they think is probably inherited or could be inherited, we encourage them to take a DNA swab. That happens, it's getting better and better, and I have a hardcore of really sort of committed ophthalmologists who send us swabs on a regular basis. But yeah, we encourage owners as well, if their dog is unfortunately diagnosed with a condition that could be inherited, to remind the ophthalmologist, could they please take a swab? If the ophthalmologist has forgotten their packet of swabs, all is not lost. Because we have plenty of other opportunities for you to request a sample kit straight from us, and I'm going to give you some of those links through the talk. So in the US, there's a very similar scheme. I found this off the internet, so although there are some details, it was on the internet, so I assume the person doesn't mind the world seeing their details. So this is slightly different, but again, sort of check boxes for different parts of the eye and any abnormalities seen, and then again bottom right another box for comments as well, and the ECVO, which is the European College of Veterinary Ophthalmology, they have a similar scheme as well, and some of the veterinary ophthalmologists in the UK use this certificate as well. So because there's all these certificates for really sort of nicely recording abnormalities that the ophthalmologists see, it's a very sort of nice system, and all of those can obviously be copied and collated in the genetics centre alongside the DNA samples, and that is really helpful. So I'm now going to talk to you about this project called CRIDE, which some of you will have heard of and some of you won't have heard of. So CRIDE stands for Consortium to Research Inherited Eye Diseases in Dogs. This was a specific project that was funded by Dogs Trust in 2019 for 2 years, but because we had a break in the middle due to COVID and the collapse of the AHT, this project has, that initial tranche of funding has sort of only quite recently come to an end, and I have put in another application to Dogs Trust that is currently under review for specific funds to keep it going. Initially, the founding members were myself and Louise. We were on the original application that we submitted to Dogs Trust, and then 6 ophthalmologists who agreed to be part of that initial consortium. They say now most of this work on a day-to-day basis is done by Kat in the lab, and the number of collaborating ophthalmologists has now gone way beyond this initial 6. So yeah, we've sort of the project has gathered momentum over the years, despite the sort of hiccups in the middle. So we're very pleased about that. And really, the project was designed to streamline the investigation of all inherited eye diseases in any breed of dog. And it does have a website, and that's at the bottom. One of the things that we have done as part of this project is to set up a database. Very aware that of all the inherited diseases in dogs, eye diseases are the ones that are the best characterised at both the sort of clinical level, but also the genetic level. There's now something approaching 100 different mutations or variants that have been published that are associated with eye diseases in the dog. Just aware it's growing all the time. So it's very difficult for the ophthalmologists to keep track of all that, as well as you as breeders. And in fact, sometimes it's easier for the breeders because you have got fewer mutations to be concerned about that's irrelevant to your breed, whereas the veterinary ophthalmologists have to keep track of everything. So we set up this database that really catalogues details of all the published eye disease mutations that have been published. Top right, there's a Getting Involved button. If you click on that, it'll take you to this page here. So this is what I was saying of you as dog owners or breeders. If you have a dog that has been diagnosed with an eye condition, or you have an older dog that's had a later in life eye exam and has come out with no abnormalities, we'd love those dogs as well. You can just click to submit a sample, and that will take you through to ordering a DNA swab kit. So just back to the database, if you would put in the search bar, so if you go up, there's a search bar here. You could put in a dog breed, and let's say you put in Labrador Retriever, you will get taken to this page here. So a list of all the variants, mutations that have been published that are associated with eye diseases in the Labrador Retriever. And then if you, let's say you click macular corneal dystrophy, that will take you through to this page here. So you can click on macular corneal dystrophy, and that will give you a description of what that disease is. OMIA, the second entry, so that's the online Mendelian inheritance in animal database that's operated and has operated for years and years out of the University of Sydney, Frank Nichols. So that's a similar database, but for all inherited diseases in all many more species other than the dog. So it's the same information is there, but it's a much bigger database. There's information there about the gene that the mutation is in, and if you click on that link, you'll find out more about that gene. But sort of importantly, the publications, any publications, and then a link to the actual publications, and then the breeds affected. So this mutation is only ever been found in a Labrador, but if it's been found in more breeds, they would also be up there as well. So this was a database that we had developed when we were at the AHT, but since we've moved to Cambridge, it's been a real advantage in that we've now been able to make that a searchable, publicly available database. So if you feel like having a look at that, it's online for anyone to do so. From that, you can also download a PDF where all the diseases are sorted by breed. The ophthalmologists find this very useful. It's quite a long document, actually. It's 26 pages long or something. So, but sometimes when they're doing their eye exams in village halls, in the Yorkshire Dales, and their Wi-Fi isn't very good and they can't actually access the database, they can have a paper copy of this to sort of remind them which variants have been found in and which diseases have been found in which breed. So I say that's available as well for anybody to download, but it is paper heavy. So just a quick bit about sample collection. So as I said, we will collect DNA. We like to make sample collection as simple as possible. So we will collect DNA from any dog of any breed diagnosed with an eye disorder that is likely to be inherited. And we take a very, we'd rather collect the sample and never use it than miss a valuable sample. So yeah, all affected dogs, regardless of the breed or disorder. And I should have said also, yeah, those older dogs that have had an eye exam and no abnormalities were found, those are also very, very useful to us as well. The samples can be collected as cheek swabs. We do ask that they're accompanied by the veterinary ophthalmologist diagnosis. So either a copy of that BVA certificate, and those of you who know, they still do a carbon copy thing, so there's usually about a rainbow of copies of each BVA certificate. So either the physical copy or a scan is fine, or a letter from the veterinary ophthalmologist that has diagnosed that dog. So we do ask that all the affected dogs come with a specialist diagnosis. So we do require that they've seen a veterinary ophthalmologist. And so down at the bottom are just some links. The top one is just our canine genetics group to request a sample from Pride, and then the bottom is actually just our general website, email address, which Brian monitors, Brian and Jim. So yeah, lots of ways in which you can get in touch with us. So since the Pride project started, we've had DNA samples from over 250 dogs that have been submitted by their owners or by ophthalmologists from 16 different countries. We've had samples from as far afield as Australia and Singapore, I think, as well as obviously a lot from the UK and Europe. We've had samples from 35 different breeds, and they've come in with all sorts of different eye conditions. And this is probably not an exhaustive list, but you know, some of the sort of old favourites there. Some of them are isolated eye diseases. Some of them are part of sort of what might be more systemic syndromes. And when we consider that, you know, we know that progressive retinal atrophy, for example, can be genetically distinct between breeds, as can forms of glaucoma. You know, we could have as many as 40 different genetic entities in this data set. So it's an incredibly rich and diverse data set that we've amassed. But just a little bit about what we do with all those samples. So the first thing we do is we screen them for all those inherited eye disease variants that are in that database. So I say close to 100 these days, because we want to know that we're not going to invest time researching something that's got a mutation that we already know about. We do that screening in batches of about 90 at one time, so that's much more cost effective. So this is definitely a research, it's not a diagnostic service. So we encourage people, if they want a DNA test result for their particular dog in an immediate time frame, then they should use a genetic testing service. This is definitely a sort of a research resource that we're amassing. So we won't necessarily screen that DNA sample for these variants. You know, we might not do that for another couple of months. So that enables us to divide our samples into two broad categories. There are dogs that we always have with the molecular diagnosis. So these are samples that we screen them for those mutations and we find a mutation that we already know about, either that's been reported in that breed before or a mutation that causes the same disease in a different breed, that we have every expectation is responsible for that dog's eye disease. So those are dogs that they're not particularly interesting or exciting, but it's useful information, and it helps us monitor how much of that mutation is still around in the population, and also whether we see eye mutations, that the variants that we found in one breed in a new breed. So it's sort of a surveillance system as well. But the samples that are, and the majority of samples fall into this category, they're without a molecular diagnosis. So we don't find any of the previous reported mutations, and so they're dogs that have a potential inherited eye disease for which there's no current explanation or variant known. So we take those samples forward for further investigation. And we have two broad approaches. So if we have multiple samples from affected dogs of the same breed with the same condition, then we will do what we call a genome-wide association study, or a GWAS. And you've probably heard us talk about those before. I suspect Sally is going to talk about a GWAS a little bit more in her talk. So I'm not going to talk about GWAS very much in this talk. But increasingly, if we have a small number of samples, so 1 or 2 or 3 dogs of a particular breed with a particular condition, we will move straight to whole genome sequencing, particularly if we think this looks like a genetically simple disease, like a monogenic progressive retinal atrophy or a lens luxation, for example. Whole genome sequencing is what it says on the tin. We sequence the whole of the genome, about 2.4 times 10 to the 9 or 2,500 million nucleotides of DNA. So a lot of you will have been familiar with our Give a Dog a Genome sequencing project that we ran when we were at the AHT. Many of your breeds contributed to that. So as part of that project, we sequenced the genomes, which sequenced 89 genomes from 77 different breeds. And obviously we still have all those data. Yeah, we were able to rescue all that data from the AHT, so we have that safely in Cambridge. So I've now got a slide that many of you will have seen before. It's a very, very simplified version of what we do with the whole genome sequence. So yeah, it's very simple, and I stand here and make it sound very, very simple. It's much more complicated, but it will give you an idea of what we do. So let's say we have a DNA sample from, let's say, a whippet with progressive retinal atrophy as a sort of hypothetical example. We sequence that dog's genome. So one little line here, but actually it's 2,500 million letters of DNA, and we actually sequence each letter about 30 times. So it's, I think, the Harry Potter series of books times 440, approximately. So it's enormous data set. And this is where being at the University of Cambridge has really been helpful to us, because the AHT in IT capabilities were, frankly, modest and a bit creaking at the seams, whereas obviously the University of Cambridge has got much, much more powerful computing facilities. So in that respect, life has got easier since we've been here. We then compare the genome sequence from our affected dog with what we call a reference genome sequence. So this is exactly what it says, a reference genome. Until a few years ago, we had one reference genome. It was a boxer called Tasha. We now have about 6, I think, different breeds. It's a mixed blessing. For various reasons, having multiple reference genomes is a good thing, but it provides much more of a headache from a bioinformatics point of view. But anyway, so yeah, essentially we line them up, and then we look for all the places in the genome where there's differences. And remember, we're looking for the variant that might be causing this whippet's progressive retinal atrophy, for example. So everywhere where we see a difference is a candidate for this disease, assuming that the reference is not a whippet and does not have this form of PRA. That will give us an enormous number of variants, that first. So then what we do is we add in more genomes. At this stage, we concentrate largely on genomes that are not the same breed and that we are pretty confident do not have this disease. And every time we see the same variant in these other genomes, we can exclude that from being the cause of this disease. And the more genomes we sequence and add in, the more the more variants we can exclude. And eventually we get to the point where we've narrowed down the search for this variant to ideally 1. Doesn't always happen like that, but that's the principle. So it's a very agnostic technique in that at this stage we're not really making any assumptions about the genes that might be involved or the type of variant. I should say also some variants are much more easy to find like this than others, but I'm not going to complicate it at this point. So there's an awful lot of variation within the canine genome. All your different breeds were evolved from grey wolves, and they've been essentially they represent a lot of variation. If you take the genomes of any 2 dogs, they'll differ in about 2 or 3 million places. If they're dogs of the same breed, they'll differ at fewer positions than if you're comparing 2 different breeds. But yeah, there's a lot of variation, and most of that variation is probably benign. It's not actually doing any harm as such, or harm that we can tell very easily. Some of it's positive. Some of the variation is stuff that you will have selected for. So dogs that are very short, we've selected over the years for short legs, or hair or big ears. That's all genetic variation that we, as breeders, have selected for. but that's not necessarily harmful. But some of that variation is deleterious or contributes to the risk of disease. And so the challenge when we're looking at all this variation within all these whole genome sequences is that we have to sort of sift out the deleterious variants from the sort of non-harmful, non-deleterious variants. So it's essentially a computational bioinformatics challenge increasingly. I've just got one slide here that talks about different types of diseases. So monogenic diseases, these are ones that are, as it says, they're caused by variants within largely a single gene. Sometimes there's additional modifying variants, but most of the disease risk comes from a single variant or a single mutation in a single gene, hence monogenic. Those variants tend, a bit of a generalization, but they tend to be in or very close to the protein coding regions of genes. And we talk about, that variant will disrupt a protein or break a protein. So they have what we call a large effect. And monogenic diseases usually segregate, within a single breed. There are some very notable exceptions. Mutation that causes PRCD, form of PRA, has now been found in tens of different breeds, and the mutation that causes primary lens luxation in many of the terrier breeds, we know that segregates in about at least 20 different breeds. So there are exceptions, but on the whole, monogenic diseases are rare, even within a breed. They tend to affect closely related subsets of a breed. And they're largely predictable in terms of pedigree analysis. If you're going to do pedigree analysis, or try to do pedigree analysis, choose a monogenic disease, because they're the types of diseases that it works better for. Complex diseases, on the other hand, they are caused by variants in more than one gene, sometimes with some environmental factors contributing as well. The variants that cause the complex diseases are not necessarily in the protein coding regions of the genes. They might be in the regulatory regions. Each individual variant associated with a complex disease might be less deleterious than a monogenic variant. And for that reason, they can sometimes become more common and maybe shared more between different breeds. So we have to keep in mind all that factors when we sift through our whole genome sequence data to find variants that we think are provocative enough to follow up. So the Kennel Club Genetics Centre has a pretty healthy, that's not the right word to use, a sizable bank of dog genomes. We've sequenced the genomes of, I think, about 311 different dogs, although it goes up and down, or goes up quite quickly. We've got at least 112 different breeds represented, and that's great because different breeds will tend to have different variations than other breeds. So the more we have got a much deeper representation of canine genome variation in 112 different breeds than if we had 300 genomes all of the same breed. But 143 of those have got an inherited, are affected with an inherited eye disease. And 79 of those have been sequenced as a direct result of the CRIDE project that I talked to you about earlier. And all of the dogs whose genomes we've sequenced for inherited eye diseases, we know are clear of all those known mutations. And again, this isn't an exhaustive list, but there's a lot of different diseases represented within that bank. So now I'm going to talk to you about one project that we have used this whole genome sequencing technology to home in on the causal disease. So it's our latest project, this disease called multi-ocular defect in the Old English sheepdog, but it could be a very similar story for a lot of other diseases in a lot of other breeds. So the Old English sheepdog is certified under the BBA eye scheme for hereditary cataract. and that's the only disease that's listed for that breed. Back in when we were running or starting the Give a Dog a Genome project, the Old English Sheepdog community participated, so they gave us some financial assistance to sequence the genome of an Old English Sheepdog that had initially been diagnosed with HC under the BVA scheme. It was that same dog was later found to have additional abnormalities that weren't picked up on that initial exam. So around about the same time, we started collaborating with 3 other veterinary ophthalmologists who had a particular interest in this condition and who had seen Old English Sheepdogs with multi-ocular defect. That was Renata, who was at the Animal Health Trust. She then accompanied James Oliver to Dick White referral, and now she's at Lumbury Park. James Oliver did his PhD with us at the Animal Health Trust, is now head of ophthalmology at Dick White referral, and Chris Dixon, who is at Veterinary Vision up in Cumbria. But they'd all had an interest in this disease, and particularly Renata, was very proactive in getting the project started. We had over the years collected DNA from other Old English Sheepdogs that had either come in with a diagnosis of cataracts or with a diagnosis of multi-ocular defect. So we had some other samples that could contribute to the project. This all sort of came together more or less. We'd started to look at this Old English Sheepdog genome when we were still at the Animal Health Trust, and then it really sort of gathered momentum when we moved to Cambridge. So we took that whole genome sequence from that one Old English Sheepdog, and we compared it at the time to 218 other dogs that were 102 breeds. As I say, our bank has been sort of gathering and growing over the years. So at the time we started this work, we had 218 genomes, and we set about looking for the variants that might explain multi-ocular defect. We made some assumptions. So we assumed it was a monogenic disease. We thought that was probably a safe assumption given on some sort of pedigree analysis that we'd done. We assumed that some of the other genomes that were of different breeds were not going to have this same variant. So on my slide, those were those control genomes that we compared this to. So this is what we call a segregation score. We're expecting to see the disease variants in only a certain subset of genomes. Because we thought it was probably a monogenic, we prioritized the variants that we thought would have a deleterious effect on the associated protein. That's what we call an effect score. And as to mode of inheritance, we sort of kept an open mind. Sometimes if we're very certain, very confident it's a recessive disease, we will look for variants that the affected dog only has 2 copies of. We won't look at variants that only have 1 copy, but with this one we sort of kept an open mind. We weren't quite sure. So variant filtering. So in those 219 genomes, we found over 28 million variants, which is a lot. So we filtered them for variants that we only found in the affected Old English Sheepdog and that were predicted to have an effect on a protein. So that's what we call filtering, and that reduced it dramatically down to 131. So it's still quite a big number, but it's tiny compared to 28 million. We then went on and looked in this thing that we call the DBVDC, which stands for Dog Biomedical Variant Database Consortium. So this is a collaboration that is a bigger set of genomes collated by Toto Lieb at the University of Bern and his co-workers, and we are members of it. So it gives us access to a much bigger data set of genomes, and then we used some extra software to predict effect, and that got us down to 22. So already it's becoming much more manageable, but don't forget we want to get down to because of these 22, only one of them likely is involved. So then we went, she says, Kat, went into the lab and actually genotyped those 22 variants in 3 more cases and 3 unaffected dogs. And we're looking at the segregation. We're looking to see the variant only in the cases and not in the unaffected dogs. And actually 21 of them did not show the right segregation. And so that got us down to 1. So at this stage, yeah, we were pretty excited. I want to say a little bit about the disease itself. So, you know, the clue is in the name, multi-ocular defect. And also bear in mind that this breed is certified under the BVA scheme for cataracts. So this chart, I don't expect you to read it all. So we have DNA from 17 Old English sheepdogs that we think have multi-ocular defect. The very right-hand column is the genotype. So the C is the mutation, and T is what we call the wild type or the reference letter of DNA at this point. So you'll see that our cases, some of them are heterozygous. And then what we've done for each case is put the age at which they were diagnosed and also which of the different abnormalities they have. So if you can't read macrophthalmos, which is an enlarged globe, vitreopathy, which is a degeneration of the vitreous or the fluid inside the eye, microphakia, which is a small lens coloboma, which is a developmental defect of the lens, and retinal detachment. So only some of our cases have all of those abnormalities. All the dogs, apart from one, have a recorded cataract. The dog that doesn't was actually quite young, and that was a dog that was actually seen at the Animal Health Trust, and we don't have access to its records anymore. So we can't be 100% certain it didn't actually have cataracts. But we think cataract is actually a consistent feature of this disease, although the type of cataract seems to also be a bit variable. And then different dogs present with a different sort of collection of these abnormalities, some of which might occur later in life. So we think actually that some of these dogs might be being diagnosed with cataracts at a fairly young age, and then might develop, for example, you know, retinal detachment later on in life. And it may be that there's a bit of a correlation between the numbers of abnormalities and the genotype. So it may be that having 2 copies of the mutation predisposes the dog to having more of these abnormalities, or maybe developing them earlier, but we really don't have enough data to be certain about that. So what do we know now? So now we conclude that this disease is associated with a variant in a gene called COL11A1. And that gene causes a similar disease in humans called Stickler syndrome. So we now think that this is a sort of a equivalent of this human disease, Stickler syndrome. There is some phenotypic variability between dogs. So not all dogs have all of these abnormalities. We're pretty certain that the mode of inheritance is dominant. And as I've said, you know, some of these homozygous dogs might have more a more severe phenotype, but we'd like more data to be certain. Cataracts, say, is a pretty consistent feature, may or may not be accompanied by other abnormalities, but those other abnormalities might appear or develop later in life, and importantly, after the age at which a dog has had a BVA exam. And some of these other abnormalities might be quite subtle and difficult to detect. Interestingly, a paper that Keith Barnett published in 1978 about cataracts in the Old English sheepdog, he referred to some of them having other abnormalities, including retinal detachment. And he said he didn't know whether that was significant or not, but as usual, Keith was was right, and they were significant. We have identified MOD affected dogs that have this variant in the UK and in the US. We haven't had access to a lot of affected dogs from many other places, but we would be interested to investigate that. But essentially, we think this variant is pretty widespread. It's on both sides of the Atlantic, at least. Our findings have been submitted to peer review and are currently under review. And once they have been published, we will certainly be discussing the implications of this with the BVA eye scheme, because we think that maybe there's a good reason to sort of change the listing of that essentially it's not hereditary cataract, it's actually multiocular defect. So that's a conversation we'll definitely be having once this has been published. So there is a DNA test available. So all of our research, once we identify variants that cause diseases, especially our monogenic diseases, we will make a DNA test available. We are, at this stage, we do advise Old English Sheepdog breeders to have this test done prior to breeding. So we do think it's a dominant disease. So all the dogs that carry that variant could pass it to their offspring, and a single copy of the variant is sufficient to cause disease, the evidence suggests. So currently this is available from canine genetic testing. Once the findings have been published and in the public domain, we would expect other genetic testing companies to offer it, but it is currently available pre-publication through CAGT. So that's the Old English Sheepdog story. As I say, we have quite a few other investigations that could progress along similar lines. But now I've just got a couple of slides to talk about a few other inherited eye disease studies that we're working on. So this year we've implemented 2 owner surveys. So these are distributed with the help of the Kennel Club and to the owners of all registered dogs of the relevant breeds. And then we also have considerable help from breed health coordinators and other breeds reps, which we're very grateful for, and makes a very successful means of gathering data from maybe the dog owners who are not members of breed clubs, for example. So as far as eye diseases, we have one going with the miniature Schnauzer communities. This is in collaboration with veterinary ophthalmologist Christine Heinrich. We're particularly interested in hereditary cataract. We're also asking questions about other eye diseases. We had a fantastic response, over 4,500 responses to that, and that data is currently being looked at by Christine. Similar survey for basset hounds. We're interested in primary closed angle glaucoma. This is a sort of continuation of James Oliver's research. We've also had a grant from the American Kennel Club Canine Health Foundation. We have had over 700 responses for that. So we're typically asking the owners of these dogs, has your dog had glaucoma? And do we have permission to contact you to get more information and maybe to collect a DNA sample? So both of these surveys are going to signpost us to dogs that have a robust diagnosis of those diseases. We should be able to collect DNA. Certainly for the mini Schnauzer cataract, we're hoping eventually to be able to refine the description of hereditary cataract as defined by the KC scheme. At the moment, it's quite variable, and we will be able to use those data and the samples that we collect for downstream genetic studies as appropriate. As I say, I would like to thank for both of those surveys and for all the other surveys that we've run this year that that are not eye diseases. Thanks to the Kennel Club for help distributing those, and to the breed health coordinators and to the other breed reps who've done a really good job, because they've all been pretty successful, yeah, they're a very useful way. Okay, so some other investigations. So this disease, retinal pigmented epithelial dystrophy, or RPED, in Cocker Spaniels. So this is a disease that used to be known as central PRA or CPRA. It's not the only only breed to be affected by it, but so whereas generalised PRA is a sort of uniform over the retina degeneration, RPED sort of starts in the middle. It's associated with a vitamin E deficiency. So we think that the primary metabolic or the primary problem is a vitamin E metabolism problem, because I gather that when these dogs are given vitamin E supplements, the degeneration of their retina slows down. We did a very successful GWAS. So I said I wasn't going to talk about GWAS, but I'm just going to show you this one. And those of you who've listened to me talk over the years will have seen these pictures before. So this is where we genotype a set of markers over the whole genome, and we're looking for patterns between the affected dogs and the unaffected dogs. So this will be a data set so this is multiple cases, I don't know, about 20 cases maybe, and a similar number of unaffected dogs. And you're looking for this over here. So where you see a big peak like this, so these are all the chromosomes along the bottom, they're color-coded, and at the side is a measure, a log scale of the association with the markers. So this is what we call the expression coined by Oliver Forman when he was in the group, a stonking peak, technical term. So this is where we're all screaming and excited, because that tells us without a shadow of a doubt which chromosome this mutation is on. We had whole genome sequences from 2 affected dogs, so we thought it was going to be, at this point, really easy to go to that chromosome, look in the genomes of those 2 affected dogs and find the mutation. Well, that was, I don't know, 8 months ago, 10 months ago. I'm very tempted to say, when I put the slide together, I was going to say that finding that mutation was challenging. As of literally 5 o'clock yesterday, we might have made a bit of a step forward. So we're hoping this is, I won't jinx it any further, but you heard it here first. So this is, yeah, certainly a condition that we feel we're certainly close. We've done 90% of the work to finding the variant that's responsible for this disease, certainly in this breed. So yeah. We're also looking at progressive retinal atrophy in English Shepherds. We have also what we call a very strong candidate. So we found a variation in the DNA that we think is causing this disease. This is not a Kennel Club registered breed, and we don't have DNA from very many cases. What would really help our cause here is, as you know, typical for all these studies, is DNA from more affected dogs, but also some DNA from some obligate carriers and some old dogs with clear eyes, so those controls. Because when we find a variant in a numerically small breed, we want to be sure it's not just something that makes an English Shepherd an English Shepherd. We want to make sure it's what's causing this disease. And so one of the ways in which we can do that is look at dogs of the same breed that don't have the disease, and we're hoping to not find that variant in those dogs. We're also looking at progressive retinal atrophy in miniature schnauzers as well. We have some clues, hints for that one, and we are currently looking at whole genome sequence from these affected dogs and some from controls. So that's another project that we're working very hard on. I think this is my probably two more slides. Just some sort of wrap up comments, really. So we set up this project called CRIES to streamline the way in which we investigate inherited diseases in different breeds. It's really gathered momentum over the last few years, and we're really hoping that we get more funding from Dogs Trust to be able to continue this and keep up the maintenance of that database. And particularly what we want to do is make the pipeline that we use to analyse all our genome sequence data a little bit different and a little bit more efficient, so that we can analyse those genomes more quickly. At the moment, it's quite a time-consuming and semi-manual process. So we're hoping that we can revamp that like everything to do with IT, you know, something that was worked and was very effective. Now we've just got more data, and we're trying to look at more and more genomes. So it needs a bit of a polish. We to do all of this work, we collaborate with a network of veterinary ophthalmologists to collect and investigate these inherited diseases, and we're very lucky. Ophthalmologists seem to be a really research engaged bunch, and so yeah, we're lucky to have some very, very good and keen collaborators. But owners can also play a really important part in submitting samples to us and raising awareness of the need for samples. So please don't underestimate your own the part that you can play, and the members of your clubs can also play. We are using whole genome sequencing increasingly to identify these novel variants, particularly for monogenic diseases, where we can start to investigate a disease with a much smaller number. When we used to always do a GWAS first, we would say, we need DNA from 12 affected dogs. And if it's a complex disease, then it's many more than that. But nowadays we can reasonably start with 1 or 2 or 3 cases as well. As I said, we're currently working our way through a lot of genome sequences. We've got tens of 60, 70 genome sequences that's on the list to look at, and we hope very soon we'll be able to go through those much more quickly than we can do presently. And we want to, the reason that there's always a bit of urgency is the sooner we can find that mutation that causes that disease, the sooner we can develop a DNA test, and the more quickly we can start to control the spread of that disease variant through the population. When we first started, those of you in the room who were part of our primary lens luxation story, for example, we'd been looking for that mutation for probably a good decade before we actually found it. Nowadays, we have a reasonable expectation of finding a disease variant from the point the first case is diagnosed to finding the mutation. It's not unreasonable that we can find it within a 24-month or even a 12-month period. And the reason we want to do that is as soon as we've got a DNA test, you can have your dogs tested and can plan matings accordingly and eliminate that disease, that variant much more quickly. So sometimes it looks as if we're studying diseases that are, why are you bothering with that? There's one affected dog being found, but there's one affected dog now, but the nature of purebred dog breeding is these populations, because you've got your popular sire effect and closed breeding populations, these monogenic disease variants can, unfortunately, become much more widespread. So that's why we like to sort of nip these in the bud. And this work that we do is, largely all about prevention, rather than cure necessarily. Okay, I've nearly finished. You'll be very pleased to know. So I just wanted to point out and embarrass a couple of people. As I say, Kat on the front has done all the work really to do with all the studies that I've talked about today. And if anybody has any real technical questions, then Kat's your person. She has all the details. Ellen is our bioinformatician who really sort of is behind the scenes looking after all these data sets, and very invaluable. Lou Pettit, who isn't here today, but she was the first person to look at that old English sheepdog whole genome sequence and had actually flagged up some of the very sort of provocative variants. So she really kicked off the whole process, and then Brian has been instrumental in collecting and collating DNA samples from for all of the studies that I've talked about today. Final slide. So I just want to say some thank yous. Thank you to the Dogs Trust, who have funded the Pride project, to the Kennel Club Charitable Trust, who supports many of the members of the Kennel Club Genetics Centre team, University of Cambridge, and particularly the Department of Veterinary Medicine. I should have said actually at the beginning that the Department of Veterinary Medicine is sponsoring today, so your lunch is on the Department of Veterinary Medicine, so thanks very much for that. And also, again, to the department for rehoming the Kennel Club Genetics Centre in the awful year that was 2020. All the veterinary ophthalmologists who collaborate with us, we couldn't really do this without us, specifically for the MOD project, James Oliver, Renata and Chris Dixon, who sent us lots of samples and lots of information about those samples. For all of our studies, really thank you to the owners who submit DNA from their dogs and information about their dogs. You know, we can't do any of this without DNA from those dogs that are sort of out there in the world. And then just the links at the bottom that I showed before, the top one if you want to request a DNA sample kit, the CRIDE one if you want to request a sample through that, and then our general Kennel Club Genetics Centre email at the bottom that will probably be answered by Brian. And I think that is me done. Probably talked for longer because I think I started earlier, but we have got, I think we're scheduled to go till 11:30, yeah. So anybody has any questions, I'm very happy to try and answer those. But if you all want to escape for tea and coffee earlier and grab me outside, we can do that as well. Yes.
Can you tell me, I observed the Old English data, can you tell me the difference between the cataracts, because you have quite a selection there? Are some congenital, which are caused by nutrition or other things? Are some inherited? How do you distinguish between each one?
So that is the challenge that they, so all those descriptions have come from the veterinary ophthalmologist.
Yeah.
So they were all descriptions largely about the type of the cataract, so where in the lens it is and sort of the appearance. So a lot of them seem to be cortical, so that's the middle of the lens. So I'm not an ophthalmologist, but my kind of simplified understanding of the lens is it's got a capsule, and it's got different layers, and it's got a nucleus, and the bit outside the nucleus but inside the capsule is called the cortex. So a lot of the cataracts are in the cortex, and they progress, but they have, you know, a slightly different appearance. So yes, so we don't. So obviously, a congenital cataract is there at birth. I think the take-home message is that this is a slightly variable cataract, and the key is it's often accompanied by other abnormalities as well. But I gather in humans with mutations in this gene, that's also a fairly variable cataract as well. So humans with mutations in the same gene that have this Stickler syndrome, their cataracts are not all absolutely identical. So some breeds have, a very characteristic type of hereditary cataract. So a lot of years ago, we found a mutation that caused hereditary cataract in Staffordshire Bull Terrier. That was always very early onset in a particular part of the lens, always progressed. A lot of breeds have a type of cataract called posterior polar subcapsular cataract that's in a particular part of the lens, so it looks like a Mercedes Y. When the ophthalmologist looks at it, it's at the back, so it's just inside. So it's at the back of the eye, hence the posterior, and it's just inside the capsule. So lots of breeds have that type of cataract. But the miniature schnauzers, on the other hand, again, quite a variable cataract, and that's why we're doing the study with mini schnauzers specifically, to try and just understand and get a bigger picture of what the cataract might look like. So for Old English sheepdogs, it's variable. We think, although there are patterns, and this is where we rely on the ophthalmologist. Often it just comes down to experience. You know, an ophthalmologist saying, I've seen this type of cataract in this breed multiple times, therefore we think that's the sort of genetic cataract. An ophthalmologist obviously can tell the difference between a nutritional cataract or a diabetic cataract. And that's why we always ask that all our inherited eye disease cases come from a veterinary ophthalmologist, because we need that robust diagnosis to say, you know, this is a diabetic cataract, or this is possibly a nutritional cataract, or some other environmental cause. So I think the Old English sheepdog story is a really good example of how it's been a true collaboration in terms of the ophthalmologists and the geneticists working together with the owners who've submitted this DNA from their dogs to try and tease apart this picture. And I'm guilty of this as a geneticist. I don't know, ophthalmologists, they just know everything. They can look at an eye, tell me exactly what's wrong with it. And they probably think, well, you know, the geneticist can look at a piece of DNA and find exactly what's the cause of mutation. The truth is none of this is an exact, science. We don't know everything at the beginning, and that's why it's called research. So I think, there are things we know now that we didn't know when we started the project, and there's things that we still aren't exactly sure of that we will, develop a better understanding for as we go on.
What about nuclear cataracts in puppies? Because it's been identified, I think I looked through some data on several of my breeds, but in Breed Terriers there's been a couple of cases of nuclear cataracts in puppies. How do they develop? What are they?
So I personally don't know. I think that would be a question for the ophthalmologist, and the way...
I've asked him.
Okay. Maybe who's he? Which ophthalmologist?
I've forgotten his name. In Nottinghamshire.
I mean, I'm not sure that. I mean, it may be that it's a new type of cataract. So I think, or that particular litter, if they were related to one, if they were part of the same litter, then there's a explanation why the dogs in that litter might have a particular type of cataract. I think it's the recurring theme is it's data collection, and when it's really not. I think the danger of the BVA scheme is that we get this idea that the particular eye diseases that particular breeds have is set in stone, and a border collie is at risk of a certain set of eye diseases, and the Labrador, they get these diseases. But DNA mutates. It's what it does. We can't stop it mutating, and so new mutations occur. And part of what we all should do is sort of be vigilant for those new conditions. So, it may be that a dog presents with a type of cataract that's not been seen before in that breed, and it might be, a novel mutation. That over time we will realise, oh yeah, this looks like an inherited condition. But if we have DNA collected, then we can have an option to sort of investigate, or they, there's another explanation for those initial cases. But again, it would be interesting to see. So I think the ophthalmologist, if it's a new type of cataract, they won't be able to say how that progresses unless they get access to those dogs and can sort of watch them over time. So that's what worked with the Staffordshire Bull Terrier cataract. Is that dogs, owners took their dogs to Keith Barnett, actually, who was able to say that, oh yeah, this always progresses, and these dogs are usually blind by the age of 3 unless they have cataract surgery. So over time, we developed a clear picture of what that cataract looked like. But at the beginning,
it's quite an odd thing. I mean, going back years ago, the ophthalmologist thought I was crazy because I brought every Shiba Inu I owned to having them eye tested before eye testing became current, because I'd sent a dog abroad and they said at the age of 6, when it had been used all over Europe, had developed cataracts. Okay. So she basically said, if you give me another dog, I won't tell anybody it's got them. I said, not only will I not give you another dog, but I'm going to tell the whole world that your dog's got it. And so we all then went on a testing mission to see if there was anything, because Shiba Inus were comparatively new at that point. Right. So I did all of mine, all my old dogs, young dogs. No one found any cataracts apart from this one dog that was 6 years old over in Holland.
So, yeah, I mean, it may be, so the genetics or the mode of inheritance of different types of cataract is different. So the Staffordshire Bull Terrier cataract was a very simple monogenic recessive disease, but that's just about, and I think now, I'm saying Finnish Lapland maybe, there is another type of cataract that's been described that is a monogenic, but most cataracts that we've investigated, we think have got a complex mode of inheritance, which means there isn't a single mutation involved. I think in general, if it's a very defined type of cataract and it progresses, and you can sort of track it in pedigrees, then maybe a monogenic. But we've put an awful lot of work into, particularly Sally, to look at different breeds and different cataracts in different breeds. And we, apart from the Staffordshire Bull Terrier, have not found a single case of a sort of monogenic type of cataract. So I think cataracts are probably, most of them, genetically complex, meaning there's not just one mutation, and there might be some environmental components as well.
So it can be environmental, it can be nutritional, it can be inherited.
Yeah, so all of those things in humans are risk factors for cataracts. So pilots apparently, are more risk of cataracts from UV exposure. Certainly, you can have nutritional cataract and diabetic cataract. So I think it is about collecting data and absolutely doing absolutely that. So when something new pops up, yeah, don't hide it. And I appreciate that can be a brave thing to do at one point, to some extent. But have dogs' eyes examined. Have dogs' eyes examined throughout their lives, because that's another thing. Some of these cataracts appear later in life. So a dog that's had a clear eye exam prior to breeding, age 1 or 2, might progress and get cataracts later on. So that's Sheila Crispin's mantra is have your oldies examined as well, because some of these eye diseases, not just cataracts but retinal degeneration, can appear later in life. And the only way to find them is to do those eye exams very often. Question back, Angie.
When you say like late onset, I think, what age?
Well,
I mean, obviously older the better, but what's the youngest old?
I think the BVA offers reduced eye examination for over 8s. And that's obviously the older the better, but 8 seems to be, I think from what we know about most diseases, most dogs will have developed their inherited disorders by 8. Not always. So retinal degeneration in setters, Gordon setters, Irish setters, can be even later than that. So it's a compromise. The later the better, but I think you get a discount once they're 8.
Yeah.
May I ask, as it's a hard thing to sell to our breeders, do they actually have to have the thing on KC, the eye test, or is it good enough just to take them to the ophthalmologist?
For the purposes of the research, I just want them to be seen by an ophthalmologist.
Right, thank you.
I just want that diagnosis from the ophthalmologist who's looked at the eyes and said, I don't see any abnormalities. So I do need some sort of, we do need some sort of record that they've seen the ophthalmologist and there were no abnormalities, but I couldn't care less whether it's a BVA eye scheme actually, or, I think, I've heard, again, Sheila Crispin say that the cheapest way to get your dog's eyes examined is to do it under the scheme. If you have just a consult with an ophthalmologist.
It's a little bit difficult. In my case, for example, I would say that over 50% of my dogs, but by the time they're between 12 and 13, have cataracts.
Yeah.
They're long-lived, which is great, but by 15, 16, when they're falling off.
So those are probably senile, old age cataracts.
Yeah.
And
so this is not significant.
I mean, I say I'm not an ophthalmologist, but people get cataracts as they get older. So an ophthalmologist will be able to look and say these are just old age cataracts.
Right.
Or sometimes it's not. What's it called? Nuclear sclerosis. That's it. So sometimes a dog's eyes will look a bit opaque when we look at it, but it's a very natural process called nuclear sclerosis. So the lens is like an onion, so it's got layers, and those layers keep increasing throughout the dog's life, so they get squashed together more. So, but again, an ophthalmologist will say this is not a cataract, it's nuclear sclerosis. And also if it's a senile cataract, an ophthalmologist will say that, be able to tell the difference as well. Jackie, I do want to keep time, so I have a couple of quick ones.
How many cases would you need to be aware of for you to conduct research into a particular breed and type of cat?
Well, so we have sequenced, one. So in amongst those whole genome sequences that we have examples of we've got it's one affected dog of one breed that's presented with a good diagnosis. And so I say we generated all those genomes because we had the funding to do it. We would probably be reluctant to say we found the cause of this disease from one case. But that genome data, it's there, it's sitting there on a computer, and we can add to it. So yeah, we've started projects with
We've also got breed-specific DNA stored as well.
Yeah, so we've, obviously healers have got this maybe possible other vitreal degeneration. So yeah, so we in amongst our genome sequences there are healers with vitreal degeneration that are in there. So you know, in theory, we always need more than that, and again we certainly need genomes or DNA from older clear dogs to compare it to. Because if we just look at the genome of one dog and we find a variation between that one dog and our 300 other dogs, we don't know whether that's something that makes, you know, a whippet or whether it's a whippet with PRA. So we need whippets without PRA to tell the difference.
When you say older dogs, would you say like these over 8?
Again, those over 8, yeah.
Yeah. So if you
unless the affected dog developed his or her disease age 6, then we might say, okay, we want our older dogs to be over 10.
Yeah, we've got no particular in our breed at the moment.
We use that over 8 as a sort of benchmark for an old dog, and then adjust it. I think I'm going to take one more question now, and then don't forget we've got that session at the end for more questions, and you can always grab me over coffee.
Glaucoma, what work is being done at the moment on glaucoma?
So we have, so our active project is the Basset Hound project. But and I think, you know, so James Oliver did a lot of work on quite a few breeds with glaucoma, closed angle glaucoma. He, again, the lack of findings of precise mutations that cause glaucoma tells us that in most breeds, probably closed angle glaucoma is a complex disease. And so investigating each breed is a bigger job. So we've concentrated on Basset. So we had some, because he did find some provocative findings. So we have got a couple of regions of the genome that we are pretty certain are involved with the development of glaucoma in Basset hounds. We got a grant from the American Kennel Club to investigate Bassets. So that's the breed that we are actively working on. But all the data and all the samples that James collected, you know, we still have those. And what we hope is that as we tease apart Basset hound glaucoma, that will give us hints as to other, maybe the genetics of other breeds. So it's you know, I never say we're not working on that anymore. We're not actively working on closed angle glaucoma in any other breeds other than Basset hound, but we still have you know, material, we have data, we have DNA samples. We will probably, you know, at this point need more funding. But you know, glaucoma is one of those diseases that would be high on our list to approach because it's difficult to treat. It's a horrible disease. It's painful. It's blinding. So essentially, I guess it's a resource issue at this point. And you know, these complex diseases, as you'll hear from Sally later, you know, they are resource intensive in terms of manpower and collecting samples. But for all of these conditions, you know, I would say encourage owners with dogs diagnosed with glaucoma to still send us those DNA samples, and we will, you know, store them safely with all their information, and they're there for when we're in a position to work more actively.